So for the last few thousands and thousands of years, humans' bodies have been wired to use food really efficiently. That's because our caveman ancestors, they were busy fighting off starvation, malnutrition. They had to stay alive long enough to have kids and ideally to raise them. Fast forward to the present day, and we are now way better at that staying alive part, at the raising and having kids. There are billions of humans on the planet now. A lot of us have way better access to food and a lot of food. So compared to our ancestors, we sort of have an oversupply or an abundance. And the way our bodies today break down that abundance of food, that's what Dr. Yi-Chun Chen is studying. She's our guest today on Researchers Under the Scope. Hi, I'm your host, Jen Cannell, and today we're at the Health Sciences Building here at the University of Saskatchewan's College of Medicine. We're at YCC's new laboratory. She's an assistant professor of anatomy, physiology, and pharmacology, and she's focused on the beta cells that live in our pancreas, the ones that make hormones such as insulin and a few others. These are the ones that have so many nutrients coming at them today that, it would totally make those caveman ancestors blush. YCC, thank you for inviting me into the lab, and thanks for being here on Researchers Under the Scope.
Thank you for having me on the show.
Well, and I understand that way back you were from Taiwan.
Yep.
And I'm wondering, where in your life did you say, okay, I think I want to become a scientist?
Okay, so I grew up in Taiwan. I finished my college education in Taiwan. But I was raised by my grandparents. So my parents, they are busy working. They are usually overseas. So I was raised by my grandparents. And my grandpa, he's a retired elementary school science teacher.
Oh.
So we would usually went out, hike and observe the geology, catch butterfly, dragonfly, and turn them into taxidermy. So I really enjoy the time spending with him. I always liked biology, math, and chemistry, so I also wanted to do something, have a career that could help other people, so that's why I wanted to get into medicine. But I also couldn't handle the stress of handling a patient's life and death, and I'm not good at memorizing the chemicals to be a pharmacist, so I thought, okay, maybe I can do a lot of lab work and become a medical laboratory scientist. So I study medical laboratory sciences and biotechnology. That's my major in undergrad. So I did a lot of lab tech work. I interned in the hospital for 6 months, and I took histology, anatomy, immunology, biochemistry, all these classes that would later on become really useful for my career. So I also went into one of the lab during the undergrad to do some research, and I was kind of hooked. So I wanted to explore more other than just working in a hospital as a medical laboratory technologist.
Oh, okay. So like the kind of folks who can come over to me and take my blood or take a sample, take it back to the lab.
Yeah, and run a bunch of tests. I was that person behind the wall running the machines.
And I mean, you could have done that for a career. It would have been a very good career.
Yeah, but the turning point is I went to my undergrad advisor's office before I graduate, and he told me, hey, you should just have a career in the hospital and find a good husband and have a family. And the feminist in me got slightly offended. So I kind of want to challenge myself. So I went to the state to pursue a master, and it was a pure cell biology master's program. So I was studying the attraction force generated by the movement of the cells.
Oh.
So it's interesting, very interesting, but I, again, wanted to do something more about human health. So I went on to pursue a PhD degree in another university. And during PhD program, I studied obesity, diabetes, and I found pancreatic beta cell interesting. So I stayed in the field of islet biology, and I moved on to postdoc. I did my postdoc in Vancouver in Canada, and I continue the line of work. And yeah, I'm still studying beta cell in my new lab.
Well, and you've done all this also, like all your background education, everything was in Mandarin.
Yeah.
And then you showed up, where were you in the United States?
I was in Connecticut for a year.
How did the adjustment to doing everything in English go?
Yeah, it was interesting. So at the beginning, I have to have a recording pen and record the lectures and listen to a lecture again in my dorm. But luckily, it's a college campus, so I have nothing else to do other than study.
So you got really good at knowing what they were saying.
I catch most of it.
That's fair. Who was it who sort of introduced you to those islet cells that live in the pancreas? Why were they intriguing?
Oh, okay. So that's a loaded question. So I think I'm biased. I think pancreatic beta cell is one of the very important cell type that control the blood glucose level because they make insulin, which is a peptide hormone to reduce blood glucose levels. And there are also other kind of peptide hormone made in the beta cell that control the satiety and body weight and other things. And other hormones like GLP-1 now is very hot. They could be potentially made by pancreatic alpha cells and have local influence on other cell type. For example, beta cell to make more insulin to lower blood glucose, or secreted in other like in the gut and again in the pancreas to control local or systemic inflammation, to suppress inflammation, to control body weight. So I think there are a lot of things we still don't know about peptide hormones, not just in the pancreas, but in the brain, in the gut. So those are fascinating for me, at least, and I want to study them more so we can potentially make more or better or effective drug to cure obesity and diabetes.
Well, because, yeah, that's the thing. I look at my cave lady ancestors and they did great. They got me here.
Yeah.
And wow, that's awesome. But now we have a whole pantry full of food. I have a fridge full of food. A lot of people across the world have all sorts of food now, and I don't have to go chase it down.
No.
So what has happened to the cells inside our body as they're dealing with all these nutrients? What question do you have to answer now?
So over-nutrient is a big thing. The cells, they are not designed to deal with that from the evolutionary point of view. So now they have to work extra hard. For example, the beta cell now need to make more insulin. And insulin is actually made as a larger precursor called proinsulin. Just like a lot of other peptide hormone, they are made as a larger peptide hormone, and they are cleaved by the enzymes in the cell, like cut them into smaller chunk. They are the biologically active hormones, and then get released into a system to regulate whatever they need to regulate. And after they are released, they also need to be cleared. So that's the thing I'm studying right now, the biosynthesis, processing, and the clearance of peptide hormone. And I'm studying beta cell peptide hormones.
And what are the hormones exactly that you're looking at?
Insulin is one. I'm still very interested in learning insulin because beta cell, they make a lot of proteins, and 1/10 of the protein made in beta cell are actually insulin.
Okay.
They make a lot of insulin.
There's little factories.
Yeah, they are. And they need to pump out the insulin. They need to process the insulin, proinsulin to mature insulin, and they need to pump out the insulin upon glucose stimulation. So they have the sensor in them to sense the glucose and make insulin, process them, and release them. And after they are released, that's a new area I want to get into, the clearance of insulin or other important peptide hormone made in the pancreas.
But you said that's 10%. What's the other 90% of the stuff they're making?
There are many things like transcription factor, the things that maintain the metabolism of the cells and maintain their survival or maintain even the structure like mitochondria, actins, and other things. Yeah.
So like the support crew.
The support crew, but their main job is to make insulin, pump out a lot of insulin, in short time. I'm talking about like quick release of insulin, which is within minutes, and when they also need to replenish the insulin pools in the beta cell, so that happen within 30 minutes. after the meal ingestion. So they start to make things. They release whatever they have first, and they start to make things and process them, and then release if they still need more.
These are really cool cells, aren't they?
Yeah, they work a lot.
They're busy.
Yeah.
Well, and I keep wandering into my kitchen and eating things, and then they have to get busy. Like, that's their job. But
Yeah.
I'm giving them kind of a lot to deal with.
Yeah,
And I'm maybe not running as much as I needed to in the past.
Yeah, so if you run and if you do other things, it would actually reduce or increase your insulin sensitivity. So your beta cell, they don't need to make as much insulin, and insulin will be much more effective on other tissue, for example, the adipose tissue or the muscle. So with the little amount of insulin they make, it can go a long way if you exercise or maintain healthy lifestyle. Go to
making
bed
them
earlier
efficient.
and make them efficient. Yeah.
There's a lot of things I'm not doing. I'm not going to bed early enough. I'm eating a lot of food, and I'm probably not exercising enough. And I'm not alone. Like, so many of us are in the same boat.
And the environmental influence, like the chemicals and other things in the environment, that's also a big thing. So a lot of researchers are working on that.
Oh.
Yeah.
So right now, what would you say the main question is that you're working on answering?
I want to understand the fundamental biology or physiology of the beta cells or the endocrine peptide hormone system, and hopefully that would help guiding the development of stem cell-derived beta cells. So we are making really good progress in Canada, actually. We have clinical trials. We are putting the stem cell-derived beta cell into patient with type 1 diabetes. So patient with type 1 diabetes, they are slightly different than patient with type 2 diabetes. Type 1 is characterized by insulin deficiency. So their immune system somehow, for unknown reason, recognize their own beta cell and kind of destroy the beta cell. So these patients, they don't have a lot of insulin-producing beta cell left, so they don't have enough insulin to regulate their blood glucose.
That's why
so usually they have the pump,
right.
Insulin pump, or they have to inject themselves insulin. That's kind of painful and really not convenient, and it's a horrible thing.
Well, and it means that you're always dependent on a pharmacy.
Yeah. And you're always thinking about, am I going high? Am I going low? It's a huge mental burden. And if you have your kid have type 1 diabetes, as a parent, you'll be worrying about whether your kid will go into ketoacidosis or hypoglycemia when they are going to sleep. So it's difficult. So for type 1 diabetes, the scientists, they are now making the beta cells so they will secrete insulin in the body. So they can better manage their type 1 diabetes without having a pump or having to inject themselves with insulin. So I want to know the fundamental beta cell biology so that the other scientists have better knowledge to make better beta cells.
Well, and with type 2 diabetes, which is
what
a lot of us acquire it, especially people who feel like they are descendants of those survivor cavemen, like they made it. But now our bodies are really packing it on.
Yeah. That's there's a lot of genetic factors behind it. It's a really complicated disease because multiple genes can control it. So there's no magic pill to cure type 2 diabetes. You can only work on personalized medicine for so much. So I think the GLP-1 drug is a huge breakthrough.
These are the Ozempics, the Wegovy's.
Yes.
Okay.
Yes. Yep. Yeah. So I would like to explore other peptide hormones and study other peptide hormones, and maybe in the future there will be other potential drugs. Yeah.
So you said these are some of the fastest moving cells too, because they've got a job to do and they've got to do it quick as these nutrients come in.
Yeah.
How do you study them? Like, how do you actually look at things in the lab?
Yeah, so in the past, people have used beta cell lines, so they immortalize a mouse or rats, and now there's also human beta cell line you can use, or I actually use a lot of rodents. I have genetically modified mouse models, so I would delete a gene, overexpress a gene, and see what's going on with the animal or just on the beta cell level, so I can. So the pancreas of a mouse is not big. It's like the size of the pinky, the first, your nail of your pinky, or the nail of your pinky. So it's not big.
No.
Yeah, and the islet cells, they are 1% of that. So you kind of took the pancreas out from the mouse and you digest them, and you get that little bowl shape. We call it islet of Langerhans. And that 1%, if you see it by your naked eye, they are like a grain of salt. So we collect those little tiny grains in mouses. Around 85% of the cells are beta cells. So we are working with a very limited amount of cells, but with the advanced technology, for example, sequencing, histology, high resolution microscope, we can really look into what's going on in the cells. And we have live cell culture experiment. We can have live cell imaging to really kind of observe the release of insulin granules. Of course, there are reporter and indicator, calcium indicator, and other things. We can visualize them better. We can't really see the release of each single insulin molecule, but we can kind of know when they are active. And do they respond to nutrient well? Do they respond to high glucose well? Or how efficient are they in making or processing these insulin molecules?
And do you test them for what could speed them up, what could slow them down, what wants to kill them, what doesn't?
Yeah, things like that. Yeah, we can add drug to them, or again, genetically delete some important gene that we think would play a role in the function or survival of the beta cells.
Are there certain genes you're looking at?
I'm studying a gene called carboxypeptidase E, which is one of the enzyme that cleaves the proinsulin into mature insulin. So I have CPE knockout mouse model, beta cell specific CPE knockout mouse model. And in the future, I would also like to delete CPE in the hypothalamus of the brain and see how brain peptides are processed by CPE and whether these brain peptides can control the satiety and the body weight of, again, in mice first.
But eventually maybe humans.
Yeah.
Humans with all those pantries and fridges full of food.
Yeah.
Well, and in terms of the work that you're doing, how does the position that you've taken on here at the University of Saskatchewan play into that? How are you liking it here?
I love it. I have really colleagues. So it's a long story. So I interview schools, and my husband and I, we decided that we want to either stay on the West Coast or in Canada. And at that time, I got lucky. I received a award from CIHR, a career transition award. So that made me strongly consider staying in Canada. And I visited Saskatchewan, and I interact with my future colleague, and they are really nice, supportive, and I can envision myself working on many interesting project with them. So I thought, this is a perfect place. And our department recently hired 6 new faculty within last 5-6 years. So it's a really renewed and energized, and it's a really good environment for early career scientists. They are like mid-career and very established scientists. They can mentor us and guide us, but we also have a group of young scientists. We are passionate and we want to work on many things. So yeah, that's why I'm here.
Oh, we are so glad to have you. And especially because I know you were on the West Coast in Vancouver for a while, and it's pretty hard to give up those beautiful mountains and sea for a smaller prairie setting, a colder one sometimes.
It's enjoyable. The winter looked really nice. At least not raining. It's just snow and it's peaceful, and you just work in your office during the winter time, so I don't see differences. And in the summer, it's really nice here. It's getting warmer, and I have a dog, and my husband and I would take the dog out on weekend, go to many places, and we enjoy the life here.
Oh, I'm so glad you chose us. Thank you.
Thanks.
And it is true, when you have a group of people that you can team up with, or you can picture, okay, where could we take this? That is one of the coolest things.
Yeah.
So down the road, like in the future, where do you see some of the work that you're doing playing into the bigger scientific picture to help humans, all these caveman descendants?
Yeah, so I think the peptide hormone, as I said, they are made as larger precursor. They get cleaved into mature peptide hormone and release. But if the beta cell, somehow they are not doing their job well, these unprocessed or partially processed peptide hormone would also get released into the bloodstream. They are not super active. For example, proinsulin only have 1/10 of the biological activity compared to insulin, so they are not active, they are just released, but we can actually detect the proinsulin level in the circulation, like just by blood drawing and doing ELISA, like analysis, like biochemical assays. So you can actually use proinsulin to mature insulin ratio as an indicator of beta cell dysfunction. So that's what I kind of work on during my postdoc. So we want to be able to predict the development of diabetes, for example, type 1 diabetes or type 2 diabetes before they are diagnosed. So the golden rule, the diagnosis criteria is usually blood glucose. But before the blood glucose comes up, the beta cell might be already working really hard. So we want to capture that early and maybe introduce earlier lifestyle intervention or earlier, like even
here's a starter kit.
Therapies.
Yeah, here's a starter kit to kickstart your immune system to make it process it better.
Yeah, or suppress your immune system in the case of type 1 diabetes, so they are not killing your beta cells so much. So when you can catch that earlier, you can actually delay the onset of diabetes by a few years, and that's has been shown to have beneficial impact in the long run. So if you can capture them early, that would actually help. So I would be interested in developing other peptide, pro-peptide hormone biomarkers, establishing them, and maybe in the future use that as a predictive biomarker of type 1 or type 2 diabetes. And I talk about the improper processing of peptide hormone in the beta cells. That is an adaptive mechanism. So they need to make more, like generate, translate, biosynthesize more molecules so they can have more chance to get processed and your body will have sufficient insulin. So they are making more peptide hormones, pro-peptide hormones, and what happened to the beta cells? So in my CPE, beta cell specific knockout mouse models, they actually have increased beta cell proliferation. So they would actually try to compensate by increase their cell mass. And I think that can only work for a little bit. But when they are so stressed, they usually decompensate. So I want to study the compensation stage and the decompensation stage. So I want to challenge my mouse with like high-fat diet or other things, or induce them to make them develop type 1 diabetes-like symptoms, and see would this process actually precipitate the disease progression, and is there a way to restore the enzyme activity so that they make mature insulin or mature other peptide hormones in the beta cells or in other islet cell types.
And that is something that could be 10, 20, 30 years down the road. We could recognize it more early.
Potentially, yeah.
Wow.
That's the hope. And I want to study how the peptide hormones are cleaned. For example, the islet amyloid polypeptide, IAPP, which is the second most abundant peptide hormone made in beta cell. Of course, the first one is insulin. So the second most abundant peptide hormone, they actually self-aggregates into islet amyloid, and they are just like A beta in your brain form these amyloid plaque that would induce brain inflammation and other nasty things. So that similar thing happened in the pancreas. And I want to study how these islet amyloid are cleared by circulating system or other system.
Right, rather than clumping up and creating those plaques, how do we turn the garbage disposal on here?
Yeah, intracellularly and extracellularly. That would be something that I would like to build on in the future.
I'm so glad that you chose the U of S to do it.
I hope I can run these experiments successfully, or I can test my hypothesis. If they are wrong, then I move on to other things.
Absolutely. Would you have any words for other women who are kind of in your same position, but maybe 10 or 15 years earlier?
Not really. I got a lot of help from my mentors. I should say that I'm really grateful for all the mentorship and support from my previous mentors or my friends. And yeah, a lot of people, my husband supported me a lot. He moved from Vancouver to Saskatchewan with me, and his family is all in Vancouver, so I owe him a lot. So yeah, for graduate student or even undergrad student, I think the life nowadays, the lifespan is so long, so you should just try whatever you want. And if you don't like it, get out.
Try something else.
Yeah.
Well, because it sounds like you've had success in the lab and you've had dismal failures probably too, right?
I have a lot of failure. I spent a long time in my postdoc, but I kind of persevere. So don't give up. If you like it, don't give up, and eventually it will work.
Congratulations again on joining us here.
Thanks.
I'm so glad you did.
Thanks.
That's YCC, Dr. Yi-Chun Chen. She's an assistant professor of anatomy, physiology, and pharmacology, and she joined the University of Saskatchewan's College of Medicine last year. She is studying islet cells and hyperglycemia. Basically, what our bodies do when they are trying to break down an excess of nutrients. You can find YCC online at ycclab.ca. Researchers Under the Scope is a presentation of the Office of the Vice Dean of Research at the University of Saskatchewan's College of Medicine. We record and produce this podcast on Treaty 6 territory, and we pay our respect to the First Nations and Métis ancestors of this place. And we reaffirm our relationship with one another. We'd sure like to thank you for listening to the show, for sharing your ears with us and being part of it. I'm your host, Jen Cannell. Thanks so much for listening to Researchers Under the Scope.